forward to talking to you a bit more about how to tune up your LNP library screening for endosomal escape and functional mRNA delivery. I'll be handing over to Juliet shortly, but I'd like to take just a few minutes to introduce her presentation. Let's get straight into it. So when we're formulating LNPs, the ultimate goal is to create a delivery vehicle for our nucleic acid cargo that is going to take it from here as it arrives to the patient to here, inside the cytoplasm of a target cell, so that the nucleic acid cargo can be released and processed, ultimately resulting in the desired outcome, such as protein expression or suppression. But the rate of success is lower than you might think. There are a lot of barriers between the dose injection and protein expression, which mean that diminishingly few injected particles actually result in protein expression. In fact, I saw this video the other day of some folks releasing 1,000 golf balls down a ski jump to try and get a hole in one, and it occurred to me that the odds of one of those balls making it into the hole has not dissimilar odds to the likelihood of an LNP successfully facilitating protein expression inside of a target cell. And that means all of those golf balls you see flying past the hole represent valuable RNA that we formulated and injected that's not fulfilling its intended purpose and ends up getting excreted by the body. Now, there are many reasons why that might be. Some particles get intercepted by the body's immune system and are broken down and degraded, the RNA degraded. Some are simply lost in circulation and never make it to the target cell or tissue type. Some end up reaching the correct tissue type, but there is no successful endocytosis, so the particles never make it inside the tissue. And even once inside the tissue, the main topic of today's talk is the fact we lose particles due to failed endosomal release, and ultimately, even when endosomal release is successful, we don't always see transfection. So each of these different stages, different barriers, results in some level of attrition of the number of particles that ultimately make it to successful protein expression. Now, the positive news is that these vaccine types and therapeutics are nonetheless demonstrating successful efficacy in patients. And so actually that means there's loads of room to optimize these formulations going forward. And if we can achieve more on-target delivery, then we can do things like lower the required dose for the patient. And that can do things like reduce negative side effects by the number of particles we're adding in, stimulating the immune system. It also means we can have more predictable treatments and ultimately improve our therapeutic efficacy. And the LNP formation itself is what really holds the key to that optimization. So the combination of lipids and RNA that we select, the way that we process them to form a particle, will ultimately result in a specific particle structure, which we can measure in various physiochemical ways. And the structure of that particle is really what is going to underpin the performance characteristics that we care about. So just looking at the lipids and RNA, we could consider the different lipid structures, the different ratios in which they could be combined, as well as the total number of components present. We're seeing in literature some examples of simpler component systems with only 3 components, and more complex ones including 5 or more. You add into that the processing methods, the fluidic mixing ratios and flow rates, the different geometries that we can use to mix those particles together that are ultimately defining that structure, which we can measure with things like size and PDI to assess the physical size and quality of our samples. We can measure the encapsulation efficiency, the stability. Ultimately, those are going to be indicators for the performance of the particle. Now, we can measure performance characteristics more directly by doing things like studies in biological solutions to establish biological stability, looking at in vitro and in vivo data, and trying to assess specific aspects of the mechanism of things like endosomal release, which Juliet's going to talk more about shortly. The other thing is that a lot of these variables are really independent, so it's often not quite as simple as just making an adjustment to one variable and seeing a trend. Multiple variables are often interlinked and interdependent and often non-linear. AI models can utilize things like neural networks, machine learning, and other techniques to gain deeper insights into multidimensional datasets, which allows us to see patterns and links between the performance characteristics we're trying to improve and optimize in our particles and the original inputs for the particle formulation and formation. These models can be used even to predict the outcomes of formulations that haven't yet been tested, which means we can direct iterative screening towards ideal particle performance characteristics even more rapidly. That being said, whilst AI models can be thought of as a bit of a black box that spits out the answers, it can't work magic, and this approach only works with enough data. And it's not just quantity of data either, quality is important too. Poor reproducibility in the process used to form particles can make it harder for the model to interpret trends accurately and reduces the efficiency of that process. It also helps to have data from different measurement techniques to provide more depth to the data set. So that'll improve the chances of identifying real correlations and being able to link them back to the relevant inputs. Now, this can be where some traditional one-by-one particle synthesis methods start to fall down. Too much hands-on time making these formulations means that generally throughput is lower, and the kinds of data sets you're able to feasibly generate are in the order of tens rather than hundreds or thousands, which compromises the ability of our AI models and tools to effectively interpret trends. Now, there are ways of automating to make larger numbers of particles more rapidly, but often those can come alongside a compromise in the consistency of the results. So introducing more process variability by trying to accelerate the rate of producing samples. Also, when it comes to then characterizing those samples, dealing with one-by-one characterization on multiple different tools is a laborious process. Measuring individually in cuvettes, DLS for particle size, as well as then doing things like ribogreen assays for our RNA concentration and encapsulation efficiency. And if you want LNP concentration, typically you need to use one-by-one particle counting approaches. Ultimately, what this results is an undersampling of the parameter space, which can lead to inaccurate or partial conclusions from our models, or even entirely missed insights. Now, our approach to solving that problem on the particle synthesis side is to combine the consistency of a microfluidic synthesis process with the sequential automation and well plate-based format in our sunscreen system. So with 96 well plates for our reagents and for our collected samples, full automation once you're running, you can get through a full 96 plate of experiments in an afternoon, with the system churning out an experiment every 3 to 4 minutes. The system uses driving fluids like ethanol and aqueous buffer to move our sample around the system, pushes those sample slugs to the mixer chip, making our LNPs, and through to a collection sample loop for dispensing into the collection plate, which means we can work with volumes down to as little as 100 microliters per input or a 400 microliter 3-to-1 experiment. The system cleans the flow path in between every experiment to ensure no cross-contamination, and that also means cleaning the glass reusable microfluidic mixer, of which we have a range of different types you can try. This same method is used in terms of pumping and mixing technology, all the way from sunscreen through to our GMP platform, Sunbather, which means that even from screening, the trends and interpretations you get from models like this are directly applicable to a process which can be used for manufacturing. As I mentioned before, the reproducibility of your process from sample to sample is key, and you can see here from data that includes 90 repeated identical formulations being run with an identical process, we have a CV of less than 1% in particle size. And even when we have bad formulations mixed in, which you're bound to experience when you're screening large numbers of formulations, the system's internal cleaning process ensures there is zero cross-contamination in the following well, and you can see that the samples run immediately after bad samples, shown in red with no PEG in the formulation, are immediately back to the same particle size and PDI as before the bad samples were run. So you can really trust that each individual sample is being run using a consistent process and is going to give you the maximum opportunity to identify trends in that data using models. You can see an example of that here in the data set shown on the top right of the slide, where we ran 32 formulations in triplicate, varying a range of different lipid composition aspects in order to identify trends. Now, this data was collected in a single run with complete walkaway operation of the instrument once you've clicked Start. You can see in the little video at the bottom examples of what an individual experiment looks like, as some dye slugs being pushed through the instrument by our driving fluids. They reach the chip and combine together in a process with our reagents. This would be making our LNPs, and the clear driving fluids continue to then push those reagent samples through the system to the collection point. In order to get all that data so quickly, we use the Stunner platform. Stunner gives us a ton of insights with just 2 microliters of sample, including the size and polydispersity of our LNPs. We can measure encapsulation efficiency with our Stunner AF variant of the instrument, which adds fluorescence. We can also measure particle concentration and detect aggregates within the samples to get better indications of sample quality. The Stunner does all of that by combining multiple different measurement techniques in a single box on a 96 well plate format. So with that 2 microliters of sample, we're able to measure UV-Vis, multi-angle rotating angle DLS, and fluorescence if you have the Stunner AF version. By combining these 3 different analytical techniques in a single box, UV-Vis is able to help us to extract total RNA concentration from our sample. The DLS, using rotating angle measurements, is able to give us particle size, particle concentration, and PDI, as well as other quality indications, all from DLS data. And with added fluorescence, we are able to then assess our free RNA concentration. Between the free RNA from fluorescence and the total RNA from UV-Vis, we have enough information to calculate and output our encapsulation efficiency. Now, UV-Vis isn't often seen as a method used to characterize the concentration of nucleic acids in the presence of LNPs, and that's mostly because of the turbidity of LNP samples. Now, Stunner has a trick up its sleeve in that we have 2 different microcuvettes from which we can measure our readings. There is a 0.7 millimeter microcuvette, in which you can see the turbidity here on the picture. And then we have a 0.1 millimeter microcuvette, which has sufficient transparency that we can get a clean UV-Vis reading of our sample. And that is what allows us, with a very short path length, to measure and unmix the RNA signal from our sample in order to accurately quantify it. Our rotating angle DLS means that we move our detector for the DLS measurement into different positions around the sample, which allows us to gain a more complete picture of sample quality. With LNPs are in a size range where smaller particles will typically provide more backscattering, whereas any aggregates present, larger particles, will typically favor forward scattering. And unless you're measuring multiple different angles, it can be hard to build a complete picture of your sample quality. So by measuring multiple different angles for each sample, we are able to calculate a 0 angle independent Z average, which gives you more insight into particle quality. Lastly, here you can see our fluorescence detection taking place in real time as we shine our blue LED laser onto our samples, and you can see the green fluorescence being returned. Now, this is on a dilution series, so that green signal is intense to start with and gradually decreases across the dilution series. This dilution series is typically what's used to provide our standard reference curve for our fluorescence measurements, and then when the system measures a sample with free RNA tagged with a small amount of ribogreen dye, we're able to then accurately measure the concentration to give the free RNA. Bring all of that together and you are able to measure size and particle concentration, total RNA concentration, and free RNA, and therefore encapsulation efficiency, all from that 2 microliters of sample on a 96 well plate format. So typically, to get all of this data A full plate is going to take around about 2 hours to run on Stunner. But if you want to just get quicker size data by measuring just a single angle, then it will take around 1 hour for a full plate. The combination of 96 well plate based formulation of LNPs with the 96 well plate based characterization of LNPs, each with fully hands-free walk-away automation, allows you to generate LNPs and characterize them with the sufficient throughput to feed an AI model with enough data, and to maintain the consistency of that data to ensure that the AI model is going to be able to give you the best interpretations possible and give you the most insights into your data sets. And that's exactly what the team at Utrecht University have been doing, and Juliet and her team have been utilizing both of these instruments in order to help understand better how endosomal escape can be optimized and improved within LNP formulations. So I'm very pleased to hand over now to Juliet.
Thank you, Ben, for your introduction, and welcome everyone to this webinar. So I'm Juliet Lefferts, and I'm a postdoctoral researcher at the Department of Pharmaceutics at Utrecht University. And during this webinar, I will tell you about the work we've been doing on the screening of LNP libraries to gain more insight into endosomal escape and functional mRNA delivery. Start off with a bit of background. Most of you will probably recognize the two researchers on the left, Professors Carico and Weissman. In 2023, they received a Nobel Prize for their work on nucleoside base modifications, as this was recognized as pivotal for the development of mRNA vaccines, which of course played a crucial role during the COVID-19 pandemic. I was also very pleased to see that they shared another major award, but then together with Professor Peter Cullis, whose pioneering work on lipid nanoparticles actually made the safe and effective delivery of this mRNA possible. Without this work on the LNPs, the mRNA-based COVID vaccines would actually not have been there. So one of the hurdles for LNP therapeutics is actually their dose-limiting toxicities when administered systemically. These toxicities can become a major challenge, as repeated or high-dose administration may trigger immune activation, liver accumulation, or inflammatory responses. These responses are especially relevant for therapies where patients require chronic or relative frequent dosing over longer periods of time, in contrast to vaccines which are typically administered only once or a few times at much lower cumulative exposure, making these toxicities generally less limiting in a vaccine setting. A major challenge for LNP therapeutics is the delivery of the therapeutic cargo at the target sites. LNPs are delivered systemically. In an optimistic scenario, only 10% of LNPs reach their target tissue. LNPs then need to be taken up via endocytosis. Once LNPs are internalized into the endolysomal pathway, the majority gets degraded in lysosomes or recycled back out of the cells. It is estimated that only 2 to 5% of LNPs can actually escape the endosomes. Finally, once LNPs have escaped endosomes, the cargo needs to be released from the LNPs into the cytosol. In the end, this leads to only a fraction of the therapeutic cargo being available. So, as I mentioned on the previous slide, endosomal escape is a major rate-limiting bottleneck for LNP delivery. As you can appreciate from these research articles I show here on this slide, we're actually not the only group hoping to gain more insights in this process and overcoming this hurdle. Over the years, there have been plenty of review articles on this topic. However, there have been no major research breakthroughs. So a main problem is that there's actually no clear understanding or consensus on the molecular mechanisms of endosomal escape. Data on this topic is quite limited actually, and sometimes even contradictory. One major hypothesis is based on membrane fusion of the LNP and endosomal membrane. So LNPs consist out of different types of lipids, one of which is the ionizable lipids. These ionizable lipids are neutral at physiological pH, but become charged in an acidic environment. So as endosomes are quite acidic, the ionizable lipids become protonated and can interact with the anionic lipids of the endosomal membrane. This interaction leads to a hexagonal phase, which you can see on the slide. And this hexagonal phase actually destabilizes the membrane, leading to the release of the LNPs from the endosomes. One way to actually study endosomal escape in vitro is by making use of the galectin-9 assay. This is actually an assay we use at the lab quite a lot. So here you have fluorescently labeled galectin-9, in our case mChair labeled galectin-9, which is stably expressed in the cells. And in normal circumstances, this gives a diffuse fluorescent pattern, as you can see on the left. But in the case of endosomal damage, beta-galactoside sugars on the inner leaflet of the endosomes get exposed, leading to the relocalization of the mChair labeled galectin-9 into the endosomes, because they bind sugars, which leads to the formation of a clear puncta pattern in the cells. And this puncta pattern can be used to study endosomal escape levels and timing. Because we hope to gain more insight into endosomal escape and transfection efficiency, we defined two research objectives. For the first, we want to see if we can develop an AI model that can predict endosomal escape efficiency or transfection efficiency for specific LNP and cell combinations. And for the second, we want to see if we can use an explainable AI model to identify specific LNP or cellular features that are responsible for these predictions, and thereby gaining deeper mechanistic insight in the biology of endosomal escape. To answer our research questions, we generated and characterized a large library of LNPs. All these LNPs are fluorescently labeled using a lipid dye, which allows for the visualization of LNP signal inside the cells. Additionally, the cells all contained a mRNA cargo encoding for a fluorescent protein, allowing for the quantification of transfection efficiency. So the generated LNPs are added to our cells. So for this we have both the stably expressing GAL9 cells, as well as cells expressing the endosomal markers Rep5 and Rep7. But today I'll mainly focus on the GAL9 cells. So cells are imaged using confocal microscopy, and these images are used for the development of our AI model. So hereby we aim to link LNP uptake or endosomal escape patterns to transfection efficiency. So the training and development of our AI model is mainly done on a specific group of control LNPs, so very well and very poorly behaving LNPs. And in addition, transfection efficiency and endosomal escape is quantified of the remaining of the LNPs from our library, which is used to both validate the AI model and to increase our understanding of LNP design and the link to endosomal escape. For the development of our LNPs, we have a specific screening workflow. Of course, we start with the synthesis of our LNPs. For the synthesis of our LNPs, we make use of the sunscreen. The sunscreen allows for the generation of large amount of LNPs at the same time. In terms of LNP composition, we decided, for now at least, to keep lipid ratios the same for LNPs, as well as NTP ratio, total flow rate, and the chip we use. After LNPs have been synthesized and dialyzed overnight, we characterize the particles, mainly based on size and polydispersity using the stunner. And finally, we perform our in vitro testing, and for this we make use of our cell lines expressing the GAL9 or the RAP constructs. So at the moment we only use the HeNAs, but in the future, and I'll come back to that, we want to use other cell types as well. And cells are imaged on our Yokogawa cell Voyager system. Here you can see example images of cells treated with MC3 control LNPs. On the right upper corner you see cherry-galactin-9 signal of cells that have not been treated with LNPs, and I hope you can appreciate that there's a clear diffuse cherry signal, whereas for the MC3 treated LNPs, and mostly for the higher concentration LNPs, you can clearly see the puncta formation after 24 hours. In addition, there's a clear DID signal of the LNP uptake pattern, and also eGFP signal is very clearly visible for these LNPs. So now I want to shortly dive into the AI model we are using. So the AI model we're using is called a variational autoencoder. This is a neural network which makes use of unsupervised learning. So in contrast to a lot of AI models, for example, the ones used for self-driving cars that make use of a ground truth that is defined by labels set by people or something else, this autoencoder does not have any additional input information next to the used images. So this model will just compress the images into a code which is described into the latent space. So this latent space describes the most important latent features that are required for the decoder to reconstruct the image again. Once the model is satisfied that the latent space contains enough and the right variables for the decoder to reproduce the input image, the model is successfully trained. Here we actually developed two different models, one based on the endosomal damage pattern using the galactin-9 signal, and one based on the LNP transfection DID signal. So in practice, this means for the training of the model that microscopy images are segmented into single cells, as you see on the left upper corner. So this is one image where individual cells are defined and used as input for the model. So these individual cells are then fed to the encoder. Images are compressed into as little features as possible, and the model is trained. So this is being done per expression pattern, so the GAL9 expression and the DID expression separately. Finally, we have 256 features that are described in the latent space. So when we want to actually use the model, we feed new single cell images to the model. It's then being run, and the single cells are compressed into the previously defined latent space. So all the 256 features that have been previously defined. So each cell is defined based on all these 256 features, and subsequently cells can be mapped using a UI model, which helps the mapping of highly dimensional data and shows clustering based on similarity between the cells. Here you can see the first results of the clustering of our cells treated with the control LNPs after 24 hours based on their galactin-9 signal, so the endosomal damage signal. As you can see, there are no very clear clusters, but there are two separate clouds. So the cloud on the left contains mostly LNPs that show poor transfection efficiency, whereas this right cloud is mostly associated with the HeNAs treated with the lipid 5-beta testosterone LNPs, which are associated with high eGFP transfection efficiency. So when we annotate these single cells, not based on their LNP treatment, but based on eGFP intensity, so based on transfection efficiency of their mRNA cargo, you see that the cells clustered on the right are mostly associated with higher eGFP intensity. Here we show similar results, however, from our TID signal, so the LNP uptake pattern compared to the GAL9 endosomal damage pattern showed on the previous slide. So compared to the clustering of the previous slide, we already see a more profound cluster for the cells treated with the lipid 5-beta tostrol LNPs. Here the same, when we annotate these cells for eGFP intensity, you can see that this cluster or cloud is mostly associated with high eGFP intensity compared to the cluster on the left. I have to add that here the single dots represent single cells based on the AI output, but eGFP intensity has been calculated on a well level, and this is something we want to still do in the future when we do annotation of each eGFP signal on single cell level. We thought initial results were actually already quite promising, but Bram Bos, one of our collaborators from the Utrecht Medical Center, spent most of his Christmas break optimizing image processing and the model development, which led to an improved AI model, of which you can see the results over here. So here we clustered again the DID signal, so the LNP uptake pattern for cells treated with different LNP formulations. And as you can see, now we have a clear, distinct cluster which is associated with cells treated with LNPs that show high transfection efficiency compared to a separate cluster which contains cells treated with LNPs that show no or barely any transfection efficiency, or cells that actually have been treated without with negative controls or LNPs without mRNA. So now that we have our model up and running, I want to focus for a bit on the LNP library that we're screening. So this is a big library screen, which we separated into a sort of primary and secondary screen. So in this primary screen, we screen about 200 LNPs, which all contain eGFP mRNA and are labeled again with the DID lipid dye. So these LNPs of the primary screen are only characterized based on size and polydispersity and added to the HeLa GAL9 cells. And we only use a 24-hour post-transfection readout in the primary screen. Then from this primary screen, we select a subset of LNPs that we want to look into further. This will be a smaller set, but we still have to decide how big or small this set is going to be. Again, these LNPs contain the eGFP mRNA and are labeled with the DID labeling. But whereas in the primary screen we just use the HeLa cells, here we only also want to use other GAL9 expressing cells. And additionally, we will also use the rep cell lines to look at endosomal markers. Additionally, LNPs of the secondary screen will be more extensively characterized, and we will also measure LNP transfection and eGFP signal at multiple time points. So regarding the composition of our LNP library, there are of course endless possibilities and changes we can make regarding the composition of the LNPs. So, as I mentioned previously, we decided to keep certain parameters such as lipid ratios, NTP ratio, total flow rate, chip size, the same for all LNPs. However, we do make use of different lipids, and we decided to focus on the helper lipids, and specifically we included mixes of helper lipids. So there is a large variety in different helper lipids based on, for example, different head group, different tail lengths, different tail saturation, etc. So in our LNP library, we decided to include a number of different helper lipids, but also use mixes of these helper lipids. So next to their different properties, we also have differences in mixability between helper lipids in our LNPs. So for now, we mainly or only actually used helper lipids at a ratio of 1:1 for our mixes. And these different helper lipid mixes are screened in combination with two different ionizable lipids, sterol lipids and PEG lipids, which leads to about 130 LNPs. And to go forward, also to screen other ionizable and PEG lipids, we will select certain helper lipids or helper lipid mixes to continue with. This table shows the characterization of the first batch of LNPs that we produced and characterized. Generally, we aim to have LNPs with a Z average of below 150 nanometers and a polydispersity of around or below 0.0 to 0.5. As you can see, most of the LNPs are below these cutoffs, and we were actually quite surprised by that the majority of the LNPs showed favorable characteristics and also were quite stable in their use. So we then treated the HeLa-Gal9 cells with these LNPs, in addition to our control LNPs, which included a positive control you can see over here, as well as a lipofectamine control for the mRNA. So as you can appreciate from this image, so every black square represents a well. You can see that there are actually multiple wells, so quite some LNP conditions that show clear eGFP signal, and maybe we actually have to quantify this eGFP signal comparable or even higher than our control LNPs. So this is actually quite promising for the helper lipid mixes going forward. This project is still, of course, ongoing, but for now I think we can draw some conclusions. First, I would like to say that the combination of the sunscreen and the standard platform actually enables for the rapid production of large numbers of different LNPs for screening purposes, and I'm not sure if you would have been able to do that with other devices, so we're very happy with that. In terms of lipids or LNP compositions, varying in the binary mixtures of the helper lipids in LNPs can have a significant effect on their transfection efficiencies. And the initial testing of the AI model that we developed suggests that both the LNP uptake pattern as well as the endosomal disruptions are good predictors for transfection efficiency of LNPs. So of course we're not completely there yet. So for our future directions and things we still want to do is of course finish the LNP primary screening and then start the secondary screening. Also for the AI model we want to further validate it, but also apply explainable AI to better understand the biology of the endosomal escape. So at the moment the AI model is still a big black box. So we have our 256 features, but we don't know what these features actually describe. So we want to get a better understanding of which features correspond with what actually in the cell, and if we can use that to further or better understand the biology of endosomal escape. And finally, we want to see if we can also use bright field images as input compared to our fluorescent images we use at the moment. And with that, I would of course also like to thank some people that have been involved in this project, mostly Bram and Sam from the UMC Utrecht, who have been mostly involved with the development of the AI model, and my colleagues at the Department of Pharmaceutics, and of course you for showing up at this webinar. Thank you very much.
Thank you both for your presentations. We will now be opening the floor for discussion. So please, if you do have any questions, remember to send us them using the Q&A feature. We do actually already have a few that have come through. So I'll start off with, I'm working with lipid polymer hybrid particles. Can I still use this method to make them?
Yeah, I can take that one. So yes, you can. So the system flow path and wetted materials are all made of very robust and chemically compatible materials, and so we can work with a few different types of solvents. And so if you're working with things like acetone for polymer particles or other types of solvents for hybrid mixtures, let us know. Reach out. We can help you out and check the chemical compatibility. But yes, the microfluidic mixers are made of glass, so we can do things like use aggressive cleaning reagents and work with a wider range of solvents. So yeah, we're very flexible in terms of the type of components that you can use to produce particles.
Great, thank you. Let's go with the AI model has now been developed fully on images from HeLa cells. Do you expect the model to work independent of cell type?
Yeah, so this is something we still have to validate, actually. Personally, I don't expect that it will be a problem. I guess when we have combined images of various cell types at the same time, it might be a problem. But when we will analyze cell types individually, I don't think it will be a problem. If it were to be a problem, I think we could adapt the model quite easily using transfer learning, so then we wouldn't need as much images for the training set. But yeah, start off with the pre-trained model and adjust it with just a couple of pictures with new cells. Yeah.
Great, thank you. We have a question which is, up to how many different lipid components can be included in these formulations made with the sunscreen system?
So that's fairly flexible. Obviously, it's quite common to use 4 components. And Julia, I think your team used 4 components for the lipid mix. Is that right?
Or 5, sometimes even. Yeah.
Sometimes 5. Yeah. So it's really up to you how many components you want to add in. It's obviously going to add some extra complexity to your potential screen. The more components you add, the more complex the kind of ratios between them and the various different combinations that you can have. But yeah, we see people working with as Julia says, 5 or even more components in their mix, or equally trying to work with simpler formulations with 2 or 3 components. So yeah, the way the system works is that these pre-mixed formulations are loaded into the wells, and then the sunscreen system is going to aspirate from each well a predefined lipid mix and run it through the system. And so you've got full flexibility within that 96 well plate to prepare all sorts of different combinations of lipids. And in terms of how the flow process works for making the particles, we have up to 3 inputs, if that was the angle the question is coming from. So we have an aqueous input, an organic solvent input with the lipids, and then we also do have a dilution input as well for producing the ethanol concentration in line.
Great, thank you. Another, can the AI model also be used to predict endosomal escape or desired formulation parameters for polymer incorporated LNPs?
Yeah, I guess so. Why not? Yeah, I shouldn't see a problem with that, actually. Yeah.
Great, thank you. How do I choose what chip to use?
So the chips that we have on offer, there's a range of different types, and so for the most part, I tend to recommend people would start out with one of our straightforward cross-mixing geometries, which come in 3 different channel width sizes. And depending on the channel width you use, you can actually use this as a lever to control things like the particle size. So generally a smaller diameter channel for a given flow rate is going to make slightly smaller particles compared to a larger channel making slightly larger particles. But yeah, the system has a lot of flexibility there that if you want to make a tweak or an adjustment to how the fluids are being mixed, to have more aggressive mixing or a preference towards more laminar flow or more turbulent mixing, then the chip is designed as a lever that you can pull to control that. So a lot of our kind of standard LNP formulations that we work with work absolutely great with a cross mixer or a T mixer.
Great, thank you. From your experiment, how do you think about size of LNP effect on transfection efficiency on in vitro condition?
So we haven't done the full analysis yet on the whole LNP library that we have screened, but I would say that if particles become really a lot bigger than 150 nanometers, that we see lower transfection efficiency. Sometimes we have particles of 150, 160, 170 nanometers, and then it's still fine. I think that's mostly due to a couple of aggregates in there, dependent on the formulations we see. But yeah, in general, transfection efficiency is lower when the particles are bigger than 150 nanometer diameter.
Great, thank you. Could you tell any NP effect on transfection and endosome escape in your AI predictive model?
Yeah, so at the moment we only included one N-to-P ratio. There were so many different parameters we could choose to adapt that we decided to keep this one the same. But I could say from previous experiments that N-to-P ratio definitely affects transfection and endosome escape. Yeah, but I think that mostly already influences size and encapsulation efficiency of the particles before. Yeah.
Great, thank you. Do experiments have to be planned in the software for sunscreen?
So the software that we use is based on having a spreadsheet that we import that contains all of the experiments you define. So you can do all of that at your own computer away from the lab, and the spreadsheet will validate your experiments as you enter them, just to make sure that all the parameters you've input are going to run successfully when you get to the system. And it'll also do things like give you an output plate layout to instruct you kind of what needs loading into which plate when you get to the lab to prep your input plates. But when you come to the software, you can simply import that spreadsheet. It will load in all the experiments and you can hit run. So in terms of the interface with the software, it's very straightforward to set up and run an experiment.
Great, thank you. What is the minimum and maximum volume of LNP that the sunscreen can process?
So the system's obviously 96 well plate based, and so the maximum we can go to is a 2 milliliter deep well plate, and so that applies to both the two inputs and the collection. So the maximum collection for a single experiment is going to be 2 milliliters. If you need more than that, then it is a viable option to run multiple repeats of a formulation and pull together those wells. As I shared during the presentation, the CV for size across repeats is very low. And means that that's a perfectly viable option for larger volumes if you want that. For the smallest volumes the system can do, each individual input channel can aspirate as little as 100 microliters of reagent. So for a normal 3-to-1 experiment, that's going to be a 400 microliter experiment. And then when you collect that, you can choose whether to collect 100% of it or take a small head and tail cut to trim the ends off of the sample before you collect it. That's user definable, and you can vary that through your experiment table as well per experiment.
How do you know if the cleaning is enough? Can you change the cleaning solutions?
Yeah, so the cleaning is automatic in between each experiment, and so it's been tested and validated by us and set to a kind of standard wash volume. But there is an option in the spreadsheet, setup spreadsheet I mentioned, where you can toggle to do either a low wash volume if you want to prioritize the system running a little faster, or a high wash volume if you want it to prioritize being a little more thorough. The cleaning solutions it's using are the driving fluids that the system uses to move your sample around the system. So that's normally ethanol and aqueous buffer. But if you're, say, switching between blocks of formulations where there's more significant changes in the formulations you're running and you want to be extra thorough, you can actually dedicate one of the wells on the input plate to cleaning solutions like surfactant-based or enzymatic cleaning solutions and have the system flush those through, followed by a wash between blocks of significantly different formulations. So you can take advantage of the system's automation in different ways to give yourself the confidence that the cleaning is sufficient. But yeah, we've had previous customers validate the cleaning with things like mass spec analysis of collected samples to ensure that there's no traces of carry over from different lipids, and have been very satisfied with that.
Great, thank you. How will you decide which LNPs from the primary screen you will include in the secondary screen?
Yeah, so actually there are multiple things we want to look at during the LNP screen, and one thing is identifying LNP compositions that enhance endosomal escape and get higher transfection efficiency. Of course, for those outcomes, it's best to select the best performing LNPs. But mostly we also want to validate the LNP model and get more insights into mechanistic features underlying transfection. And so I think for that purpose, it's the best to get a whole range of responses actually. So we will quantify mRNA transfection, so eGFP levels for these NMPs, but also endosomal escape based on the GAL9 pattern we see, so the number of puncta or the puncta area. And based on that, we can sort of divide LNP compositions based on how they perform on transfection and on endosomal escape, and then we will select from all these sets NMPs. So also not LNPs that aren't performing very well and NMPs that are performing well. Yeah.
Great, thank you. As a bit of a follow-on, can you draw any possible early learning from potential factors impacting on endosomal escape?
That's something we actually really can't answer at this moment yet. So this is something we will need to investigate in the follow-up studies using also the rep cell lines, and also really looking at what is underlying the transfection and the endosomal escape. So no, we haven't been able to look at that yet.
How does the cytotoxicity factor include into your LNP selection criteria for the secondary screen?
So we actually also just count cells based on nuclei. So that's something we will take into account if some particles show high toxicity. We will probably not continue with those particles, and we will measure this based on cell viability, based on the cell count. Yeah.
And then I think we've got one more. What imaging resolution is required for the AI model to perform reliably?
Yeah, so that's something, that's an interesting question. So that's something we still want to validate. So at this time, we make images at a 60 times magnification. I think we might could go down to 20 times, but you need to see all the individual the fluorescent pattern. So I would go to a high magnification. Yeah.
Great, thank you. I think we're just about there with the questions. Don't see any more. Lovely. Well, thank you everyone for joining. If you have any queries following this webinar, please do get in touch with the presenters using the contact information that will be shared. We will also email ways to get in touch along with the on-demand recording in the coming weeks. So please do look out for this in your inbox. Thanks again to Juliet and Ben for their presentation. That draws the webinar to an end. Thank you everyone, and have a good evening.
Thanks.